Hello everyone, welcome to our seminar on the urinary system. I will be explaining you guys about the kidney. So let's start by its functional unit, which is called as the nephron. The nephron consists of all of these structures over here. So firstly, we have this area which is called as the renal corpuscle, and the renal corpuscle continues into proximal convoluted tubules. This entire part over here is called as your proximal convoluted tubule. The proximal convoluted tubule continues to form this loop, which is called as the loop of Henle. And the loop of Henle later continues as the distal convoluted tubule, which then continues as the collecting duct. There's two types of nephrons. We have the cortical nephrons and the juxtamedullary nephrons. The cortical nephrons are mostly predominant, and there's about like 80% of the nephrons in our body are cortical nephrons, while as the juxtamedullary nephrons are only 20%. And the difference, the major difference in them is that cortical nephrons have a shorter loop of Henle, whereas the juxtamedullary have a longer loop of Henle. And this line over here just tells you what part of it of the nephron lies in the cortex of the kidney, whereas what lies in the medulla. So this part above the line is what you will find in the cortex, and this part below is what is going to be found in the medulla. Just to summarize what I said, the cortex of the kidney contains a renal corpuscle, proximal convoluted tubule, and the distal convoluted tubule, whereas the medulla consists of the Henle's loop and the collecting ducts. So firstly, we have the renal corpuscle. Renal corpuscle has this outer structure, which is called as the Bowman's capsule. And the Bowman's capsule has two layers, as you can see here. The first layer, or the outer layer, is called as the parietal layer, and it is lined by a simple squamous epithelium, whereas the inner layer, it is lined by these special cells, which are called as podocytes. Moving on to the inner area of the renal corpuscle, it is made up of something called as the glomerulus. So the glomerulus is a tuft of fenestrated capillaries, like I had mentioned earlier. It has two parts to it, the afferent arteriole and the efferent arteriole. The afferent arteriole is from where the blood enters in, gets filtered over here, and then it leaves through the efferent arteriole. If you get confused between the terms afferent and efferent, remember it this way, that exit stands for E and efferent also stands for E. So efferent is something that's going to be carrying a substance away or out of the body because it's exiting the body. So over here, the efferent arteriole is carrying the blood out of the renal corpuscle. So the part where the afferent and efferent arterioles are located, this area is called as the vascular pole, and the area right opposite to it, like this part, it is called as the urinary pole, and this is where the primary urine is stored. So all of the space over here, which you can see, this is where the primary urine is stored. So next we have the tubules. There's two types, like I said, proximal and distal. The proximal convoluted tubule, it has a very irregular and a really, really narrow lumen. That's because the nephrocytes of the proximal convoluted tubules have a brush border which like is facing towards the lumen. And it's made up of simple columnar cells, so it's like a thick structure which you will see in your slide, and these are also like really really eosinophilic or red stained, and this is because of the abundance of mitochondria. In the proximal convoluted tubule, you will not see a lot of nuclei because the size of the proximal convoluted tubule is really really large. So when you like make a cross section of it to form the slide of the kidney, you will not like you cannot see a lot of nuclei which are caught up in the cross section. And the boundaries between the cells of the convoluted tubules, they are not very prominent, so you pretty much can't see them in both proximal and the distal one. And now the distal one differs from the proximal in the fact that its entire structure is a little bit different. It has a larger lumen, which is more wider than the proximal convoluted one, and this is due to the fact that the nephrocytes of the distal convoluted tubule do not have a brush border. They are lined by simple cuboidal cells instead of simple columnar cells, and you can clearly see like the circular nuclei of them. They also have a lot more nuclei compared to the proximal ones, and the cytoplasm is also significantly lighter. Uh, there's this one particular place where the distal convoluted tubule is called as the macula densa, and that's like right besides the vascular pole of the glomerulus, when there is this one distal convoluted tubule which is present like right besides it, and this part of it particularly senses the concentration of urine, so this is called as the macula densa. Next, we have the Henle's loop. So this part onwards, I'm going to be talking about the medulla. Uh, Henle's loop, it is a very tiny tubule and it has a really small lumen. It has two types of limbs. It has a thin limb and a thick limb. The thin limb, it is made up of the simple squamous epithelium, and the nuclei are really protruding. And there's something called as vasa recta, which are vessels which accompany the thin limb. Then we have the thick limb. The thick limb has simple cuboidal epithelium, and you can clearly see the distinct differentiation between these two in the slide. Next, we have the collecting tubule. The collecting tubules have a really large lumen, and they're made up of simple cuboidal to simple columnar cells. And you can also see the boundary between these cells distinctly, so you will definitely not get confused between collecting tubules and any other sorts of tubules which you see. The function of these tubules is to see that 5% of the kidney's reabsorption of water and sodium happens in them. They have two types of cells. There are the intercalated cells and the principal cells. The intercalated cells, they have a lot of cell organelles such as the mitochondria and the smooth endoplasmic reticulum, and the surface, which is like pointing towards the lumen of them, is covered by something called as microplicae, and these structures are basically just they could be either microvilli or protrusions of the plasma membrane forming like something similar to a microvilli. So they are just called as microplicae, and the function of the intercalated cells is maintaining acid-base homeostasis, which basically means like they determine how the kidney is supposed to act if it's undergoing alkalosis or acidosis. Next, we have the principal cells. These cells mostly predominate in the collecting tubule, and they do not have as many organelles as the intercalated cells do. There's two hormones which play a role here, aldosterone and ADH, and these ones are which control water resorption. So now there's some important points which you should be knowing. Firstly, the blood-brain barrier. Like I said earlier, podocytes are in charge of this mostly, but there's three layers to it. First, we have the fenestrated endothelium of the barrier. So these ensure only like a few particles which go through into the glomerulus. They do not allow every single particle from the blood to go in because that's the point of fenestrations, right? They have these openings of only specific size, so they allow only those particular particles to enter through. So that is going to be our first layer of the blood-urine barrier. Then we have the glomerular basal lamina, and this layer is fused with the basal lamina of the podocytes. So this does not allow large proteins to pass through it. Lastly, we have the podocyte pedicles, and they also have like a really small diameter, so they allow only very few particles to go through. So this is how the blood and the urine is kept separate from each other, so they don't allow the mixing of it. Next, we have something called as the juxtatubular macrophages, and these they are as essential in our body because macrophages remove harmful substances, right? So these ones remove something called as sediment particles, which are present in the urine, and this is basically helps our body to prevent the formation of kidney stones. Moving on, how does the kidney determine the concentration of the urine? So there are two ways in which it does that. Firstly, we have the loop of Henle, and then we have the vasa recta. The loop of Henle, the first part of it, which goes down from the proximal convoluted tubule, is the descending limb because it's going down into the medulla, right? So this part is going to be permeable to water, and then it curves, and then it starts going up again into the cortex, which is called as the ascending limb, which is impermeable to water. And then we have the vasa recta, which basically prevents something called as the medullary gradient. So let me just explain you this part a little bit more in detail and in simpler terms. Uh, the descending limb of the vasa recta, as blood moves down to the medulla, it enters like a more saltier environment because the medulla has a slightly saltier environment than the cortex. So to balance the salty environment, water leaves the blood, and the salts enter into the blood. When I say salts, I mean like sodium and chlorine ions. So these enter into the blood in the descending limb of the vasa recta to balance out the amount of salt which is present in the environment of the cortex. And this makes the blood in the descending limb more concentrated. Then after it like forms a loop and then it starts going back up again when it's part of the ascending limb, uh, again, it enters into the environment which is like less salty because the cortex does not have that much salt as the medulla. So now the blood is salter than its surroundings. So the water moves back into the blood and the salts, all of them move back out. So this restores the blood's normal concentration. So this is how the concentration of urine is basically determined, uh, depending upon what part of the kidney it is, and if it's in the cortex or if it's in the medulla, and then it decides how much water needs to go in or how much like ions and stuff need to go in or out. Okay, so theory is mostly done. So now let's see how the slide looks like. This is what the slide of the kidney looks like. It's really pale, but once you zoom in into the cortex, we are in the cortex right now. Okay, so like this really dark stained part, this is going to be the cortex, and this light stained part is the medulla. So when we zoom in into the cortex, we see like a lot of glomeruli which are present here. Like see here, like well, renal corpuscle is here, one more, one more. There's like so many of these. So I will zoom in a bit more to explain you guys a few details. So okay, let's take this one for example. You can see that there's two layers, the outer layer, parietal layer, and then the visceral layer. Between this, we have the space in which the urine is stored. And this area over here, this is going to be our vascular pole, because usually like vessels and stuff, the afferent and the efferent artery will enter over here. And then this will, this part is going to be our urinary pole. Then when we go aside and we talk about the tubules, this one, for example, let's see this one. This is going to be a proximal convoluted tubule because firstly, there's not many nuclei which you can see. Secondly, like it's really, really thick and the lumen is very narrow. Like you can barely even see a lumen, and it's like very reddish as well. So this is your proximal convoluted tubule. So is this and this one and this one and this one and this one. There's a lot of these. Then when we talk about this one over here, for example, you can see firstly the lumen is really large. It's clear for that matter, and you can see like a lot of nuclei. So this is going to be your distal convoluted tubule, and so is this one, and so is this one. So if you can see like there's a clear difference between the lumen of a PCT and the lumen of the DCT, because there's so much space over here and there's barely anything here. So this is how you identify between the proximal and the distal convoluted tubules. Now, when we move on into the medulla and you zoom in, you only see like a bunch of these tiny loop of Henle segments. So the difference between thin and thick segments is that, okay, let's see, this one, for example, there's this one I would call a thin segment of the loop of Henle, and so is this is also a thin segment. This is a thin segment. This one would be a thin segment, and so would this be. But the thick segments are like significantly thicker. You can see this one, this is way thicker than this one because this one's like barely a line, but this one, it has a simple cuboidal epithelium going on. And so is this one. This one would be a thick segment, and this would also be a thick segment of the loop of Henle. But this big structure over here, this is a collecting duct or a collecting tubule, because firstly, it's way larger than any of these small segments of the loop of Henle. This one has a really large lumen. So does this one. So these both are the collecting tubules. And you can also see that there, you can see like each and every cell, you can see the boundaries between them. So, and also they have like a circular nuclei. The cells are between simple columnar to simple cuboidal. So yeah, this one and this one would be a collecting tubule. And that's it, honestly, for the slide of kidneys.
All right, guys. So now we'll talk about ureter. Um, so ureter has three parts again. We got tunica mucosa, tunica muscularis, and then we have the tunica adventitia. So when we talk about the tunica mucosa, it has many longitudinal folds which lead it to having, stellate type shape. And when we talk about the epithelium, it's lined by urothelium. And as you remember by the first lecture itself, the first seminar, urothelium is the transitional epithelium, which is also referred to as pseudostratified cuboidal epithelium. Then when we talk about the lamina propria mucosa, it is made up of loose connective tissue and it forms the core of the mucosal folds. When we talk about the tunica muscularis, it is made up of two layers. It has the inner longitudinal layer and the outer circular layer. And in the lower part of the ureter, there's a third outer longitudinal layer, which is continuous with the bladder's muscles. When we talk about the tunica adventitia, it contains fat, blood vessels, nerves, as well as it is made up of loose connective tissue. Look at the slide of ureter. As you can see, these are all the folds which lead it to having the lumen having the stellate type of shape. And when we talk about, the epithelium, as I said, it is urothelium, pseudostratified cuboidal epithelium. And it's cuboidal because you can see the shape of the cells is, circular more than, elongated. And when we talk about this is the lamina propria mucosa made up of loose connective tissue. And these are the muscle layers, and it's a muscle because especially it's a smooth muscle because the nucleus is flattened present at the bottom. So this would be the inner longitudinal smooth muscle layer, and that's the outer longitudinal smooth muscle layer. And then here you have the tunica adventitia. As you can see, there's a lot of connective tissue. These are blood vessels. So that's about it for ureter. A really simple slide, and most likely it will be the similar shape in your slide test as well. Now, when we move on to urinary bladder, urinary bladder, it's again three parts. We have the tunica mucosa, tunica muscularis, and then tunica adventitia. When we talk about the tunica mucosa, again it contains thin folds and it is lined with transitional epithelium called urothelium. And when we talk about the lamina propria mucosa, again loose connective tissue which forms the core of these folds. When we talk about the tunica muscularis, it is really thick layer and it is really prominent on the slide. And the smooth muscle layer is known as the detrusor muscle, and it is organized into three layers. These are the inner longitudinal layer, the middle circular layer, and the outer longitudinal or the oblique layer. And tunica adventitia, again, loose connective tissue, vessels, as well as nerves. So when we're looking at the slide for urinary bladder, as you can see, all of this is, muscle. This is muscle, this is muscle, this is muscle. So this is the detrusor muscle. How do you know it's a muscle again? As you can see, the nucleus is flattened, present at the bottom. Now, when we talked about the epithelium, again, it was urothelium, same as ureter. As you can see, the cells at the top, they're more circular. This makes it having the transitional epithelium, or the pseudostratified cuboidal epithelium. And then when we look at the bottom, loose connective tissue, which forms the core of the fold, this is called the lamina propria mucosa. Yes, this is the muscle layer, inner longitudinal, middle circular, outer oblique layer. And here again, you have the tunica adventitia. You can see a really small part of it. It is the loose connective tissue, vessels, nerves. And here we have another slide of the urinary bladder. So remember, for urinary bladder, you can have two types of slide, one with the longitudinal cut and then this one with the with another type of cut where you can see the lumen. Now, main difference you will notice is that the lumen of urinary bladder is way bigger and it does not have a stellate shape like we saw with ureter. I will show you guys again. So you can see the ureter has a really small lumen as well as it has stellate shaped. When we talk about the urinary bladder, it has a really big lumen, and yeah, it does not have a stellate shape. So here again, large lumen, which is a characteristic feature compared to the ureter, which has a stellate lumen, as I said. Uh, and then again, same with the epithelium. You can see the epithelium more clearly here than the previous slide. So the stratified cuboidal epithelium, lamina propria mucosa, uh, inner longitudinal layer, outer circular, middle circular layer, and then outer oblique layer. So that was about the urinary bladder. Now, when we move on to the parts of male urethra, there's not a specific slide for it for now for the urinary system, but for the next test, which will be the male and female reproductive system, then we will explain you guys the slide. But theory can come in this micro test about these two slides, the male urethra and the female urethra. So when you talk about the parts of the male urethra, it is divided into intramural part, prostatic part, membranous part, spongy part, as well as fossa navicularis. So when we talk, and these different parts are lined by different types of epithelium. We have the intramural part, which passes through the wall of the bladder, and it is lined by the transitional epithelium. And when we talk about the prostatic part, again, passes through the prostatic gland, and this is lined with the transitional epithelium. When we talk about the membranous part, passes through the urogenital diaphragm, and it is lined by stratified columnar epithelium. This part is here. And then we have the spongy part, which is the pendulous part here. It is located in the corpus spongiosum of the penis, and it has the stratified columnar epithelium. And then we have the fossa navicularis, which is the terminal wide opening, which is here. And this is lined by stratified squamous non-keratinized epithelium. This was an image I found online, so just go with what's written on the slide. These are stratified columnar epithelium, not pseudostratified, and stratified squamous non-keratinized epithelium. When we talk about the features of the histological slide for the male urethra, you cannot see all the parts. This is the cavernous part of the male urethra. So you can see the epithelium would be the stratified columnar epithelium, and the lamina propria mucosa contains intraepithelial mucus glands as well as loose connective tissue. These are called Litter's glands. And it is surrounded by corpus spongiosum, which is an erectile tissue, again, which will be explained in the next a seminar. And when we talk about a skeletal muscle which is located in the male urethra, that would be the urethral sphincter. Uh, Now, when we talk about the female urethra, there is no slide for this, but you need to know the theory for this. So when we talk about the tunica mucosa, the epithelium, it varies by regions. We have the transitional epithelium, which is near the bladder. Then we have the stratified columnar epithelium in the intermediate zone, and then we have the stratified squamous non-keratinized epithelium, which is the most common throughout the female urethra. When we talk about the mucus glands, there are two types of mucus glands present. We have the endoepithelial mucus glands of Morgagni, which are found in the epithelial lining, and then we have the paraurethral mucus glands located in the lamina propria mucosa. When we talk about the another feature of the lamina propria, we have venous plexus. These are called thin-walled veins. And when we talk about the tunica muscularis, it has the inner longitudinal smooth muscle layer and the outer circular smooth muscle layer. And then we have the tunica adventitia. I'll just show you guys the slides for the male urethra, just to give you a small idea about it, right? So you can see this is the corpus cavernous, this is the cavernous part, and then you have the corpus spongiosum, which is the most important part of the slide. Here you can see the different types of glands. So here, as you can see, these are the paraurethral glands or the Litter's glands, and since they are branching out from the epithelium, they are called the branched mucus glands. And when we essentially talk about the different parts of these glands, this is the secretory portion, as you know from your basic histology, which is the light part, and then you have the duct part, which is the darker part. Okay, and anything else? Yeah, so these are the exoepithelial, or also called the branched paraurethral glands. These are called Litter's glands. Both of these glands are mucus glands, as you know. And here you can see the epithelium, which is the no. And another type of glands present is the intraepithelial mucus glands. These are called the Morgagni glands. Anything else in this slide? I don't think so. Again, as we said, cavernous part, these are the cavernous spaces which are lined by endothelium and filled with blood. And when we look at the next slide, we'll have a better view for the other parts. Here, as we can see, this is the other the fossa navicularis. You can see this part. This part is called the fossa navicularis. We have the stratified squamous non-keratinized epithelium here on the lining on the top, and below this we also have a thin layer of lamina propria mucosa. Uh, and here we have the cavernous spaces which are filled with blood. These are called blood cells basically. So this is just blood and nothing else. So yeah, this is for the male urethra.